Hi, everybody. I'm Celeste Fraser with the Watershed Coalition, Estes Valley Watershed Coalition. I know most of you, I'm sure. It's great to have you. I wanted to mention, before we introduce our speaker, Jeff Ravage, that we are going to have a party celebrating all the work that we've done for the past, 11 years now, since the Estes Valley Watershed first began in 2015. And that was a response to the flood of 2013. So we're going to have a lot of stories. I see some people who are here are going to talk about those days. We're going to have a lot of stories and a lot of food and drink. Please come to the party. It is Thursday night, February 5th from 4:30 to 7. We'd love to see you there. And Jeff Ravage, I just watched a video of you. It's very interesting. He's doing excellent work, and it's all about mushrooms and carbon-free destruction of the debris. Jeff, take it away.
All right. Hi, everyone. My name is Jeff Ravage. I'm the Chief Science Officer for Coalitions and Collaboratives, an NGO here in Colorado that works closely with fire, forest mitigation, training, mitigation professionals, and dealing with social issues, and also running grant programs that help fund smaller nonprofits that are in the space, such as the Estes Valley Watershed Coalition. Um, I'm going to go ahead and share my screen now. And, oh, sorry, technical difficulty, shared the wrong screen. Yeah, let's try that. All right, can everybody see? I think, raise your hand if you can see it, because I think everybody's muted. All right, cool. Well, hi. Welcome to our talk, Applications of Fungal Decomposers for Biomass Remediation and Ecological Restoration. I'm going to go over uh, in the next 30 or 40 minutes about a dozen years of research and application in dealing with the use of wood rotting fungus to deal with some of our um, ecological issues. So we're going to start with an introduction to cold fire. Cold fire is what we call our process. Uh, it is the use of targeted, trained fungi to rapidly decompose biomass. And as you'll see as we go ahead, there are several different distinct applications of this. So to find out why we call it cold fire, we're going to talk about briefly what is hot fire. And hot fire, which we all are familiar with, is actually two chemical processes. The first one is pyrolysis. This is the thermal decomposition of organic materials, and it involves a change of chemical composition and a physical phase change. We call this in thermodynamics a second order phase change, which means it occurs slowly, although in some cases it seems like it's rather rapid. It's endothermic, which means it requires exterior heat. Basically, what is going on in this process is all of the molecules in the atoms are shaking and starting to fall apart and change. Uh, the cellulose, which is one of the main components of the wood, is breaking down into methane, into phenols. It's becoming tars and releasing these exotic organic compounds. But nothing really happens until the second chemical reaction, and that's called combustion. And that's the one we all know and we all like watching when we sit around the campfire. It's the thermal transformation, the final transformation, and it is a chemical composition phase transformation and what we call a first order transformation, meaning that it occurs instantly. It recurs instantly and it releases a lot of energy. Releases that energy as heat. That heat becomes the preheating for more pyrolysis. The pyrolysis reaches its flash point and combusts, and it goes on and on again. The cellulose is reduced to carbon dioxide, carbon monoxide. There's actually going to be methane released that some of it will combust and some of it will go in the atmosphere because there's just only so much oxygen for all of these different chemical reactions that are occurring willy-nilly all over the place and in a rapid succession. It also releases heat and it releases ash. Cold fire is fungal decay. It is a second order phase transformation, which means that it takes some time, but it's an enzymatic transformation of these organic materials, and it occurs at ambient temperatures. So whatever the temperature is within a range, I mean, you can get freezing and it will stop. You can get too hot, it will stop. It is an irreversible change, and it is chemically almost indistinguishable from fire. The cellulose is, but with some actually helpful differences. The cellulose is converted to carbon dioxide. It's converted to water vapor. It's converted to energy, but not heat. And that energy is used to build chitin, which is very similar to the cellulose molecule, and that's what the mushroom is made out of. It's made out of chitin, which is basically the material that crab shells are made out of, or insect shells. It relieves sugars and it relieves ash. So we call it cold fire because it is definitely doing the same thing as fire, and it is taking the biomass or the material out of the fire cycle and putting it into the soil cycle where it is safe, where the nutrients are preserved and many nutrients are destroyed in a wildfire by the fire. And it builds the nutrients and the richness of the area rather than destroying it like a catastrophic fire does. So we began experimenting. We were like, at the time, I was at the Coalition for the Upper South Platte, which is the organization that Coalitions of Collaboratives grew out of. It was a small place-based ecological organization. We did a lot with, fire and defensible space and helping out neighbors with forestry issues. We ran several slash sites, so people would bring in all their branches when they were doing mitigation on their own property, and we would chip them. And we found ourselves every year with, a few thousand tons of chips, and it was really hard to get rid of. We didn't know what to do with it. We were also doing mitigation projects, and one of the big costs of doing mitigation to try and clear out the forest and reduce the intensity of these fires is, what do you do with all the slash that's generated? What do you do with all the small unmerchable trees, which in Colorado is most of them? And we were talking about it, and we were like, well, let's try mushrooms. And since I had taken my ecology and I had grown mushrooms before, I was like, let's give it a try. I was skeptical. I mean, it seems good on paper, but things that seem good on paper do not always work out in real life, especially because our main concern was not only does this work, but does this work in any way that could possibly be useful? So our basic method is to collect and propagate, and we train them. We train the mushrooms that we've collected from an area where we want to use them on the material we want them to eat so that they recognize it as a food source. Obviously, wood rotting mushrooms will eat wood, but it might not immediately recognize either a different species of wood or wood in a different form, like chips, as opposed to a tree trunk. So the training is how we get them to identify the material that we want them to decompose and how to do it rapidly and efficiently. Our experiments have always been involved with, once we've got the mushroom trained, we want to have as little disturbance, and little fanfare about how we put it in. We have specific methods of putting in the ground. We do not break apart our blocks because the block is kind of like the mothership of the fungus. It has the energy and the capacity to produce a all of the antibiotics, and fungi is where we get most of our antibiotics, and all of the enzymes required to break down and take over this pile of wood. So that's what we did. Our first experiment was 10 1 by 1 meter plots. 5 were inoculated with a trained native oyster, and 5 were controls. And we had various treatments. We had just wood chips with some nutrients poured on it. We put some uh, humic acid and some fish emulsion fertilizer, and then we did the same thing with burlap, or jute matting on the top, to see whether or not putting jute matting would uh, hold in more moisture and make it work better. So jute matting, nutrients, no nutrients, no jute matting, nutrients, no nutrients. Uh, and so there were 2 that ended up with, anyway, there are 5 of them. All right. And we let them run for 5 years. And in that 5 years, what we saw was slightly over 50% loss in bed depth, in mass. We saw, if you look down at here where it says stages of decay, those stages are stages that we came up with, because the stages of decay that's used in the literature for forestry did not really apply to what we were doing. So a stage 1 wood chip is basically a wood chip as it would come directly out of the wood chipper. Stage 2 is a wood chip that's involved with mycelia. So the mycelia is growing through it, it's active. And stage 3 is the finished product. It's a pretty much compost. And that's kind of what we call it, is stage 3 compost. And as you can see over the seasons, the percentage, you can see the stage 2 coming in, and then stage 3 coming in. And in the end, stage 3 is highly predominant. One thing you have to think about in these graphs, they look like they're the same size, but by season 5, the amount of material in the mushroom here, yeah, I can see my pointer. The amount of material in this mushroom bed is half what it was over here. So this amount of that isn't digested is very little, and it looks like there's more pure wood chips, but those pure wood chips are the ones sitting on the top of the pile. They're exposed to the atmosphere. They're too dry to be involved. They're kind of like the cover shell, but it looks like a higher percentage of the depth of the pile, because it is a higher percentage of the depth of the pile, but it's the same depth as we had on day 1 of the dry chip on the surface. But in the controls, after 5 years, we had only around a third of the depth loss and no compost created. And it was only beginning to get into stage 2, which means the natural fungi were only beginning to find it after 5 years. And from the literature, we really expected these wood piles to last between 20 and 60 years if they were left on their own in the woods. Nothing was done to them. Fungi would get them. Fungi would do exactly what we're doing. We're trying to do it faster. We're trying to do it more controlled. We're trying to do it in a manner where we can convince people who are doing forestry, do this, don't burn the piles. Save the fuel, save the emissions, and save the risk of starting another wildfire because you're burning wood piles out in the forest. So we want to look at what is the chemistry of this compost we've created. Is it similar to what would naturally be produced, or are we producing some exotic material that's going to throw our forests out of whack? But when we look at our burying compost, which was our experiment, compared to the organic A horizon of forest soil, which is a fancy way of saying duff, it compares very nicely. The one thing we did notice, though, from the beginning is the organic matter, which is carbon, was nearly twice as much as it was in the natural duff in the ground. And so we thought that's that's something to look into. And that's something we've been looking into ever since. Our first huge replication that we actually did all the monitoring and we published was at the Chatfield Farms of Denver Botanic Gardens. Our one windrow of wood chips was 210 feet in length. It was 81 tons of mixed ponderosa and lodgepole wood chips. You can see the before and the after. This is 3 years of decomposition. In that 3 years of decomposition, oh, and this is just kind of how we designed it. And this drawing is today still kind of what we base all of our uh, inoculations on, putting spawn on alternating sides of long windrows or piles so that they can grow together and infiltrate the entire pile. In our 3 years, we went from 142.6 cubic yards in volume to 32.8 cubic yards in volume. We went from approximately 81 tons to 7 tons of material. We had an average of 77% reduction in volume and an 85% reduction in mass. In this particular experiment, we had 4 different species of mushrooms. We had uh, a Ganoderma, which is a relative of reishi, but it's one that grows here in Colorado. We had a turkey tail, we had an oyster, and we had Flammulina velutypes, which is known as enoki in the grocery store. And so we were also trying to see if there was a big difference in uh, the digestive patterns and efficiencies of these different wood rotters. And there were some slight ones. We're not going to go into that here. But what we are going to go into here is what happened to the carbon. So, since we had our eyes on it, we had samples from the beginning and we had samples to the end. What we see at the beginning of our experiment is the carbon profile of that pile. So, basically, we had 44 tons of non-carbon and 30 tons, or yeah, 22 tons of carbon. We're looking at it at a molecular level. Actually, this is carbon, not carbon. At the end, uh, we had, and I'm just like looking at the graph. We had 5 tons of not carbon and 2 tons of carbon. And without getting too deeply into the weeds, what we had is we had about 10% of all the carbon that was here at the beginning is still left at the end. Right now, a lot of people are saying the penultimate preserver of carbon of biomass in the forest is biochar. Biochar preserves 13%. So we're right about online with biochar, but we don't need $1 million pieces of equipment with lots of people to run them. And risking, what's that again? When you're burning things in the wood, risking starting another forest fire. What we look here, TCO2e is a fancy way of saying tons of carbon dioxide or its equivalent. You may have heard that methane is, 20 to 80 times more detrimental to global warming as carbon dioxide. So when you count a molecule of methane, you give it, say if you had a ton of methane, you would say was 80 tons carbon dioxide equivalent. And there are other gases that are nitrous oxides, other things that we monitor. So what we have here is the fungal emissions, and then we have the baseline emissions. This gap here is all potential, and it is 1, 2, 3, 4, 5, 6, 7. It's about, I think it was about 8 tons of carbon dioxide that was held out of the atmosphere more than would have been held out by a baseline method of dealing with this wood pile. So we are doing more experiments with that. We're working on ending a big one right now. What we saw that was really interesting was we saw mass evaporating because when the mushroom eats it and uses it, it exhales carbon dioxide and water vapor, and that's all that's in cellulose. It's carbon, hydrogen, oxygen. So it's completely clean, and it's evaporating, and it's evaporating at about a 50-50 amount. 50% is carbon dioxide, 50% is water that goes out of it. But it is mass disappearing, and it's just really kind of cool if you're into that sort of thing. So the implication of this is that if we can use mushrooms to rebuild soils, rather than throwing all of this away, we have the added benefit that we are sequestering carbon into the soil in a form where it's actually stable. Our model has always been the boreal forest, which is the forests that surround the Arctic Circle on the planet, and British Columbia, and Siberia. Those forests, they hold a massive amount of carbon in them. And the reason they do is there's a large amount of lignin in that soil, and lignin is very stable. And the lignin is in the soil because of mushrooms. So we were like, what if we could do that? What if we could uh, enhance soil uh, in a natural way to actually sequester carbon and then let the forest continue pulling carbon out of the atmosphere? We've created a planetary machine for removing carbon dioxide from the atmosphere. And that's exactly what we need to have. There's a possibly apocryphal story, which means it may be malarkey, but I've heard it said that Elon Musk offered $10 million to anybody who could develop a machine that could pull carbon dioxide out of the atmosphere and bind it so that it could be sequestered. And somebody replied to him on Twitter, "Well, that's a tree. Where do you want to send my $10 million to?" And he took the post down. So, but that's what we're trying to do. We are advocating what's called nature-based solutions, using the ecology as it's a technology in order to reverse the damage that our misunderstanding. And that's really all it is. We've misunderstood how the planetary system works, and we've misunderstood our place in it. We kind of think that we're in the boss, and we most definitely are not. And if we don't have a little bit of humility and look to nature for how to solve these problems, then our problems are going to be worse. I'm not going to say it's completely doom and gloom, but we have a better solution to offer. So if we can sequester carbon, which is the first part, so we take carbon and we've nabbed it away from the atmosphere, but then for it to be considered removal, we have to keep it out of the atmosphere for 100 years, and that's where the lignin and the soil complex works for us. Carbon removal, right now, we're sticking, what is it? We're sticking like 35 gigatons of carbon dioxide in the atmosphere every year. And it's built up. I think I'm trying to remember because this was part of another presentation that had the numbers, so I didn't have to remember them. But there's something like about 500 gigatons of carbon that we have to remove from the atmosphere to get back to where we were in like 1900. And where we were in 1900 at like 278 parts per million of carbon dioxide atmosphere had been stable for 800,000 years. It had been that, and then all of a sudden they're doing it. So if somebody tells you there's natural variability, you can say yes, every million years there's natural variability. But what we want to do is we want to pull it out, and in order to do that, we've got to pull tons of carbon dioxide at the minimum every year out of the atmosphere before the turn of the century to start the tipping point back in the direction of having a functioning atmospheric system. Right now, we're removing zero point zero 0.02, well, 0.002 gigatons of carbon dioxide a year. And if you look at it, the target range, it says BECCS. That's basically fuels. So they're saying that if we create a fuel plant and take our waste biomass and turn it into biofuel, that we are removing. And it's not, because it's going to go right back in the atmosphere. But that's the best they have. Other novel CDR, this little tiny slit, is the type of stuff we're working on. Actual natural solutions. We have to turn this around, and we can. But the first step in doing it is understanding what's going on and understand what we can do about it. And the big thing I think that everyone needs to understand is that this is understandable, that it's not rocket science really, and that we all can contribute in small ways, and small ways magnified by all 8 billion of us, we can turn this around. And methods like the fungal decay of waste biomass is one way we're going to do this. So we always wanted to go to the boreal forest, so we did. We went to British Columbia in order to replicate this, and also because I wanted to get a few boxes of soil from there to see what exactly their soil looked like, since I've been reading about it in textbooks for so long. And we were selected as finalists in an X Prize competition called the Fire Grand Challenge. The Fire Grand Challenge was an international competition of novel technologies that had to do with the fire cycle and how we interact and try and mitigate our problems with catastrophic fire. We will be going on the 26th of this month to Oakland to find out if we won. We very well might. I think we're probably going to be on the podium, but I really hope that we win, just in order to give some more legitimacy to these types of methods, because there are other scientists working on stuff like this all over, and they're the ones that get the very tiny crumbs of funding and the very tiny crumbs of public awareness. The ones that are getting all of it are computer technology, AI. Everybody's like, "Oh, AI is so great." It isn't, but it does consume money at an heretofore unknown rate. So that's what we did there. Also in 2025, we did a project in Estes Park for this little group called the Estes Valley Watershed Coalition. And we treated, what was it, about 50.7 cubic yards of material. This is the layout of, we had 6 piles in 2 different locations. And this is basically a rundown of what our volume was, and how many spawn bags we put into it, what was the rate of seeding. This will help us down the road as they decay in order to see how much is left. And it's also, we always like to keep track of the rate of seeding. We've had success with the rate of seedings up to 400 to 1, which means that you have 1 pound of spawn material to 400 pounds of material to decompose. And knowing the most effective and most efficient rate of seeding under different circumstances is an economic thing, because we want this to be economic as well. Because society always recognizes the economy. If we can do it cheaper, it doesn't matter whether we're ecologically positive. People will adopt it. I wish it was otherwise, but that's kind of the way it is. And we think It already is, but we want to be better. We want to get better. So our next steps are we're starting a large mine reclamation project, which unfortunately I can't show you yet, but we'll be able to show you the design plans, and I could just tell you it's going to be really cool and really big. We're also going to hopefully start this year our post-fire inoculations, where we're going to go in and inoculate these dead standing trees and see if we can turn them back into soil in 3 to 5 years. Because a lot of times my first fire restoration project was the Hayman fire, and there are still dead standing trees in the Hayman. I've helped plant over 200,000 trees in there, seen a survival of about 20% of them. And I would really love to have a way to accelerate turning those dead trees. And they're a hazard too. You can't go out there and hike around because one of those could fall on your head at any minute at the slightest gust of wind. So if we could drop them down, turn them in the soil, and then we'd have something for our new trees to eat, because we'd be recycling those nutrients, and we should have better success with replanting. And that's what I'm going to be doing the next 20 years. So that's what we're doing right now. And I had to just put it in chimeric transformation because we're also playing around with the potentials of actually 3D printing mycelium. So that's what we're up to. There's some compost sitting in a pile of wood chips. And if anybody has any questions, I will stop sharing and open it up to the floor. I can't hear you, Celeste. You're muted.
Okay. In three to
Ah,
5 years, do the mushrooms die, or are they propagating themselves throughout this process?
they actually live. So it's the same organism. The mushroom itself is merely a reproductive organ. The actual living entity is the mycelium that's inside the pile of the tree, the whatever. So yes, we call it overwintering. So they survive in there. We have not yet seen them be able to, spread spores and start growing, on the next wood pile over yet. And that's part of the reason why you want to use these blocks to spawn, because everybody's like going, they think, well, do you use spores to do this? Uh, Spores are any given mushroom can release 1 trillion spores, and the chances are that none of them will create a living organism, because a spore is not a seed. A spore is more like an egg and a sperm, and you have to get two complementary ones, just not two of any of them. Two complementary ones have to find each other out in the big wild world within you know, a couple of inches of each other and be able to grow and join together in order to become what's called a diploid, which is a full genetic complement individual that is a fertile adult that can then grow and start eating and eventually reproduce.
Hmm.
Okay, so Rachel had asked, what temperature range? Well, whatever the ambient temperatures where we're at are. Uh, Obviously, they will stop. Uh, they'll stop working probably in the low 40s Fahrenheit, and then they will also stop working in the mid 90s Fahrenheit. But it's going to take 180 degrees to kill them Fahrenheit, and it's probably going to take almost, I can't even say that, 'cause I was just telling Willem we just got a cryogenic storage unit, and we can actually store these things at negative 84 degrees Fahrenheit, and revive them. So that doesn't kill them. But
How do you,
generally, they exist in nature, and their metabolism speeds up or slows down depending upon what it is. Obviously, they're more active in the summer, spring, and fall, and they go dormant in the winter, and
yeah,
uh, that's what they're up to.
how do you scale up? Have you thought about what happens in another decade? How do you scale up?
We're in the process of scaling up, and how we're scaling up is we have this facility, and we are working with other people who are mycologists or people who have mushroom farms across the country. Right now, we're in 2026, we're going to be working in projects in Washington State, in Oregon, in California, in Tennessee, and in Colorado that we know of so far. So we work with the people that are on the site 1,000 miles away. We get there, we get the mushrooms. They either collect them or we go and collect them, and we get their wood and we take them in our lab and we train them, and we send them back a couple hundred pounds of mother spawn, and they expand them into the bags that go into the ground and put them into uh, their own projects. Uh, so it's a decentralized economic system where small communities actually get the majority of the economic benefit of this. But we get enough. We likened it when we had to build our business model in the Fire Grant Challenge to Coca-Cola. We're making syrup, and then all of our distributors all across America are doing the final product, and they're benefiting. But there's going to be so much activity that will benefit too. And our goal was always to have a stable place where we can do even more research because we're messing with plants right now too. Okay.
How do you train mushrooms?
Generational conditioning. That's about all I can say in a open up. But yeah, it's look at it this way. These organisms have been unchanged for hundreds of millions and some of them a billion years. During that time, they've eaten a lot of things. And the soup of chemicals required to digest those things are stored in their DNA. They're just not turned on. The field of epigenetics, which has just been exploding in the last few years, is looking at how different characteristics can be turned on and off by environmental stimuli. That's what we do. We stimuli them. And yes, is mushroom training scalable for large scale? Yes, it's exactly what we want to do. We want to be doing thousands and thousands of acres a year because otherwise, what's the point? It's like I said earlier, this is really cool, but if we can't make it work at the scale of our problems, that it's just another curiosity. And we don't think this is a curiosity. We think this is a viable technology. Is there a certain depth you need for a chip pile? Um, Chips cannot be spread loosely across the ground. Although, ask me in 4 more years what you do with those, because we have an idea. But for this particular technique, we want them 2, 3 feet, even 4 feet deep, because wood rotting mushrooms don't do well with soil. They can't send their mycelia across soil. It has to be a wood to wood contact because they grew up inside of a log for the most part, most of them. So that's what they know and that's their habit. Um, so the chip spread across the ground mastication is a huge issue, and there are ideas on how to do that. And we're going to, not in 26, but in 27, we're working on designing a project with Jefferson County Open Space where we're going to try some methods of doing that very thing. But it's one of those things where it's going to take us a while. We have to be really careful. Uh, and we always have to be careful with what we're doing because the idea of even though these are mushrooms that are native mushrooms, we're putting an organism back into the environment. And, like Colorado, Fish and Wildlife would tell you, if you raise that raccoon, you're not putting that raccoon back in the wild. It's your raccoon now. Um, and it's the same thing with any organism. You have to be very careful about how you do it, and you have to monitor it. You have to watch and see what's going on. Because, yeah, like one of the other questions is, we do not want a large group of wood-eating mushrooms uh, taking down our forests. And there are different types of wood-eating mushrooms. There are ones that only eat dead materials. We call them saprophytes. The ones that we worry about are parasites. And there are some saprophytes that are facultative parasites, which means give them a chance, they're going to be an opportunist about it. We don't want to use a species like that either. The largest organism on the planet is the humongous fungus in Oregon, and it is, I would say it's actually a facultative parasite, but it's doing real good at it. It's had some real success because it covers about 2,400 acres. So, but its main method of survival is wood rotting. So what it does is it it rots the wood and it eats it and it does its thing. And then when it runs out of trees to eat, it becomes parasitic and it kills a bunch of trees and then it eats them. And then when they're done with that, it switches back again and kills some more. And it's really interesting. There's been some interesting studies done on it. They haven't really asked some of the wild questions I want to know, but I will eventually get there. And because it's very interesting. It's why is this organism doing this there and it's not doing it anywhere else? I mean, kind of, but there's no humongous fungus junior in California. There's no cousin of humongous fungus in Colorado. It just doesn't, and the fungus and the mushrooms are here. They're simple honey mushrooms. Uh, they're ubiquitous across the Western United States, but in this one place, they're doing something unique, and it's definitely one of those things, head scratchers. Is
If you
Rocky?
look, sorry,
Oh no, go ahead.
if you look at acreage such as the national park here in Colorado, and I don't know how many trees they have to deal with, fallen trees, dead trees, etc. But how can you get a supply of mushrooms to deal with that expanse of acreage? Where do the mushrooms
Well,
come from?
it's all in what we call amplification. So when you're dealing with mushroom spawn, when you have the mycelium growing in a healthy manner and are able to cultivate it, you can turn. I think the standard thing is you can turn 1 tablespoon into about 10,000 tons in 1 year,
Hmm.
and
One
each of those
tablespoon
10
of what?
of living mycelium.
Okay. Oh,
And each one of those 10,000 tons can then rot 400 times as much. So
hmm.
that's 4 million tons per tablespoon. That's the cool thing about fungi. They're really, really good at eating and growing. And that's why it works. It wouldn't work very well if they weren't. Fortunately, they're not picky eaters. So, it's Rocky Mountain National Park aware of this project? Kinda. I asked them for a permit a couple years ago to collect there for this project because I actually want to get mushrooms for the Estes Park project. And they came back and said, well, the national park considers anything that lives on the park their property. So if you were to take it, they would remain their property in perpetuity, and they would have to have some sort of ownership in your company or corporation because there's too
No,
much potential for profit being made off of this. And so I left it at that. I figured they just need some more time, a little more education. We need to get a little bit bigger. We'll go back and they'll see what we've done. I tried to tell them, we're a nonprofit. We're not doing this to sell money. And any mushroom we take from you couldn't be used more than 100 miles away from you. It's not like we can turn this into, you know, something that we can spread around the world because that's not our philosophy. That's the status quo mentality of how you do things. And that's why we've degraded this planet so much. Uh, every ecosystem is unique. It's a community, and they're all inter, intertwined with each other. And these communities are as small as our human communities are and as specialized. So,
I have a question.
yeah.
Um, the project that we did here in Estes, I I'm on the board of the Watershed Coalition, so I'm familiar with the fact we did it, but I'm not very familiar with the status of it, what the outcome was. But I seem to recall the first step didn't really take. Is that correct?
Not to my knowledge. Um, you're not expected, it's not done. You're not expected it to be done for 3 to 5 years.
Right. Okay. So that's
Yeah,
why I'm asking. What we did was I don't know what you call it, inoculate
right.
the
So,
pile.
so we initiated the process. It's in nature's hands right now. So
Oh, okay.
what we do right now is we monitor it, we watch it, we measure it. That's why we did all the measurements and everything, so we can measure it and see, because it's incrementally. It's obviously going too slow for you to see with your eye, but when it's done, it's like that picture of that big pile at Chatfield. You can see the difference. So we're just waiting. Yeah.
So will like this year, will you go out and see how much growth there has occurred out there and how it's
Some will.
progressing?
Some will. Absolutely. Yeah. Not till spring, but yeah.
Okay,
Yeah.
cool.
Christian had a question about gasoline powered chippers. And Christian, the wood chips is not the only method we can do with this. Uh, I didn't throw that in. If you have full log trees, we can do plug inoculations, and we're doing those right now in Woodland Park, uh, where we're working in areas that not because it's a wilderness, but because the slope and terrain are too extreme, and they have to do treatments that they call lop and scatter, where you don't take any of the material off of the landscape because it's impractical to do so. But we can go in and we use plug spawn inoculation. We use little dowel rods. It's the same way the Japanese have been growing shiitake for about 1,000 years. And so we go in and we inoculate the logs themselves. And that's exactly what we're doing in post fire in those trees is we're using dowel inoculation. But we have some really cool Japanese guns that inject it in so that we could do it rapidly because you got to do a lot of them. Uh, and of course, they're Japanese because they're created by shiitake farmers. And we thank them for making it because they're really cool.
Hey, Jeff, I think there were a couple of questions on there too about what would are you do you have any thoughts about whether or not Rocky or the US Forest Service might be willing to adopt this instead of burn-in slash piles?
Yeah, but there's legal constraints on them as well as what we call institutional inertia, which means we will keep doing what we've been doing because that's what we're due. Uh, but no, actually, one of the I was telling you about large grant that we're applying for. One part of that grant is working with the U.S. Forest Service and the BLM on creating protocols on NEPA, on making sure that we have protocols that are accepted by the National Environmental Protection Act, and then yeah, and then separate. And it all has to do, of course, with publishing things, and it's slow. I mean, we've been doing this for 12 years. We've got 2 articles published. That's the speed of science, and and we'll just keep going, and it will grow because there are other people doing this too. And there's other stuff in our British Columbia project is actually an experiment, and it's being monitored by PhDs, and it will be published in Canada because they want to let the Crown uh, Forestry Department know about this as well. So the U.S. Forest Service is far more familiar with this than the National Park Service, because you would think that they would be close, but they're not really. The National Park Service is a fiefdom to their own.
Yeah. And the State Forest Service is interested too. They came out when we did our inoculation. I think
Yes,
we had 4 or 5 foresters that came out, so that was kind of cool.
but
There's
yeah, all of them just need to see this more. They need more validation, more verification, and that's our job is to give it to them. So,
and there was another question in here too. What would a large scale project such as this look like in a forest setting? I think your chat build project was pretty large, right?
right. Well, but yeah. So we're designing a project to be hopefully adopted by Colorado Springs Utility on Pikes Peak, and they're doing 1,000 acres of mitigation. And our design basically has when you go in and you're doing these large scale mitigation projects, you create temporary roads. They call them skid trails, so that all the disturbance is kept to a small area. You don't want to be driving over everything if you don't have to. Um, so our prescription is as they go in and thin, all of the the tops, the limbs, and the unmerchable stuff gets stacked on the side, either side of those skid trails. And then the last movement is a large chipper on tracks goes through, and you chip all that material right onto the skid trail. And we cover every skid trail just like arterial system over the mountain, and we inoculate that, and then gravity will eventually distribute our compost into the soil, and having that pile on top of the area that's most compacted and most devegetated helps restore that little area of ground, those roads, so that the roads will naturally regenerate in and of themselves. And by doing this, we're not also, it's really important to realize is we're not reintroducing anything. And a lot of the methods we've used in prior times to try and rehabilitate these skid trails in these areas where compaction has occurred has the unintended consequence of like bringing in a lot of weeds, or bringing in something that wasn't there before. And where we have done it, we haven't seen any change in the vegetative composition of the area. So we're hoping that'll be just, another cherry on top of the milkshake. So for doing this on a residential property with the smallest pile size you could use, I wouldn't I wouldn't want to do it on less than 2 or 3 tons. So that would be a pile that would be, 3 feet by 4 feet by 10 feet. I wouldn't want to, but that's it's totally doable if you live on a forested property and you're doing some of your own mitigation work. Uh, but if you're in town and you're just trimming your trees, I don't think this would be the way to do it. Uh, hopefully, eventually, and some municipalities like Boulder are collecting that type of material in their recycling program and using
Jeff,
it
I think
to
Everything in
compost.
you're
leaves?
frozen. You were frozen there. Do you mind repeating that?
Oh no, can you hear me now? Am I
Yes.
unfrozen?
Yes.
Okay. So yeah, I was just saying that if you're in a residential property and just have tree trimmings, I don't think it would be appropriate. But if you got forest and you're producing some real chips, then it's feasible.
So anybody who does this has to create wood chips first.
Yeah,
You can't just have a burn pile and start with that. You have to chip it up for this to begin seriously.
correct.
Okay.
We've inoculated burn piles. We've had some of them actually get eaten by the mushrooms, but it's not reliable, and it's because there's too much air. The fuel is too, you know, it's too dispersed. We
Mhm.
want wood on wood contact, uh, and mass. Oh, so Joan said she had a 3 by 4 pile that she took one of the leftover bags. So yeah, let's see what happens.
Any other questions from anybody?
Well, this has been very interesting, Jeff. Thanks
All
for
right.
making this possible.
Well, thanks for having me. And we might be seeing you all soon. We'll probably be doing some more piles in the Estes Park area.
I
And
hope so.
and if you happen to have friends that are in the park, put in a good word for us.
Sure. Well, and all of you remember the celebration on February 5th, Thursday night, 4 to 7.